Okay, so we have been working on a couple of proteins, and I want to give you an update on some of the things we've been doing. So we've been trying to track down a mutant and trying to pack up a punch. So the people who have done the bulk of this work are Byung-Hak Ha, who's a very talented postdoc in my lab, and Matt Davis, who is a very talented grad student in Yossi's lab. So, track down a mutant. We've been collaborating with Ruth Halaban and the Yale Spore in skin cancer in this massive exome sequencing study that was published in Nature Genetics a month ago. One of the goals here was to understand and to see if there were any other mutations that were associated with melanoma. So it's well known that BRAF and NRAS are associated with 40 to 50% of melanomas and maybe 15 to 25% of melanomas. The Yale Spore discovered that RAC1 is actually mutated in about 9% of sun-exposed melanomas. And it's a mutation that occurs at codon 29, so it's a proline to serine mutation. So this was published in Nature Genetics last month. So what we've been trying to do is to understand what the functional and molecular effects of this mutation are on RAC1. So let's just stand back and with a brief overview of what RAC1 is. So RAC1 is a small GTPase, a small G protein. And these are very elegant, very beautiful switch proteins. They cycle between an off state, an inactive state that's bound to GDP, and an on state or an active state that's bound to GTP. In the active state, the protein is able to bind to downstream effectors, and that causes an increase in downstream signaling, or is compatible with downstream signaling. So to cycle between this off state and the on state, the GTPase needs to release GDP and bind to GTP to get to hit. And the way it does that is it's aided in release of GDP by the guanine nucleotide exchange factors. To get back from the active state to the off state, so to turn the signaling off, there's actually a different mechanism. And the mechanism is the hydrolysis of GTP. So the gamma phosphate is released, and this is catalyzed or helped along by GTPase activating proteins. So we are a structure biology lab. We look at crystal structures predominantly of proteins. So we're interested in understand what the molecular level mechanisms of how these things work. What are the changes that occur between the off state and the on state in the small GTPase? And at the most simple level, there are conformational changes that occur between when the molecule is bound to GDP and compared to when the molecule is bound to GTP. And the conformational changes occur in these regions called the switch 1 and switch 2 region. So if you superpose the inactive and the active state on top of one another, you can see actually there's a conformational change in this red region and also in the blue region. This conformational change may look insignificant, but what it means is that the GTP-bound protein can actually bind to downstream effectors. So this is a complex between RAC and PAK crib domain. And some of you may be able to see that the conformation of the RAC of the GTPase in the active state actually allows binding of GTPase, whereas the GDP-loaded form actually maybe isn't able to bind to the downstream effector. So, question we've been asking first of all, and customarily addressed in Ruth's paper, was does RAC1 P295 display an increase in downstream signaling? So the upshot is yes, it does. When you look at pull-downs of GTP-loaded wild-type RAC1 compared to the mutated RAC1, you see an increase in the binding to the crib domain of PAK. Ruth also did a number of assays in melanocytes and showed that the P295 mutation is associated with increased melanocyte proliferation, increased melanocyte migration, and increase in ERK activation. So the answer to this is yes, RAC1 P295 does display an increase in downstream signaling. So the next question that we've been addressing here, and that I'd like to show you, is what are the molecular mechanisms for P295 activation? And the asterisk here is the secondary question: does it activate in the same way as mutations in RAS? So we'll know that RAS is, the RAS genes are mutated in somewhere on the order of 30% of cancers, and the significant proportion of those mutations are at codon 12 and at codon 61. So what do those do to GTPase cycling? These mutations at codon 12 and codon 61 put a big traffic light at the active state, and they prevent GTP hydrolysis and lock the GTPase in the GTP-loaded state. So the GTPase is unable to be activated by GTPase activated protein. So the big traffic lights are stopped, and you have an increase in downstream signaling because the GTPase can't get back to the off conformation. So what are the molecular mechanisms of the P295 mutation in RAC? So the location of P295 is on this switch 1 loop. So switch 1 loop is in green, switch 2 is in blue, the phosphate binding loop is in pink, and bound GTP is shown here. So proline 29 is close to the guanine moiety in GTP, but it's structurally distal from codons 12 and codon 61. So codons 12 and codon 61 cluster close to the gamma phosphate of GTP. So we wanted to ask the questions, is this an active GTPase, but how? Is it potentially because of loss of hydrolysis? Is it gap insensitive, GTPase defective? One would intuitively think maybe that's not the case. We wanted to show that. Is there altered effector binding? That's potentially something that could be interesting. Or could it be a fast cycler? So could it be a GTPase that has increased cycling, which has been shown to be oncogenic for CDC42? So we solved crystal structures of a bunch of different RAC mutants, and these crystal structures for wild type, for the proline 29S mutation, for a phenylalanine 28 to leucine mutation, which is a known fast cycler, and for the Q61L mutation, which is one of these mutations that has no activity. The upshot from the crystal structures is that the proline 29S has an altered switch 1 conformation compared to wild type, F28 and Q61, and this is actually quite different to most structures of row family GTPases. The second point from the crystal structures is that the interactions between the switch 1 loop and the GTP are actually different between P29S and F28L. So we wanted to look at the effects of this mutation on nucleotide exchange. So one of the assays we did was to use a fluorescent GTP loading assay. So the way this assay is done is GTP-loaded GTPase is incubated with a fluorescently tagged GTP that's non-hydrolyzable. So as the GTPase releases GDP, so as it inherently releases GDP through its natural breathing, the fluorescently tagged GTP is able to bind. What that does is it causes an increase in fluorescence upon stimulation. So you can see here for the wild-type protein, there's very, very slow exchange of GDP for GTP. Once this protein is actually incubated with EDTA, there's a dramatic increase in the switching between the GDP-loaded and the fluorescently tagged GTP-loaded form, and that's because EDTA chelates magnesium, pulls the magnesium out, so that's the glue that keeps the nucleotide bound. EDTA pulls magnesium away and allows the cycling to occur. Proline 29S is different. The mutation proline 29S is not stably GDP-loaded. It actually cycles right from the get-go, and so the GTPase is bound to GDP a lot weaker, and it breathes a lot more and allows the fluorescently tagged GTP to bind. So what this says, we think, is that the P29S mutation can cycle even without the addition of a guanine nucleotide exchange factor. We then wanted to double check that this mutation has lost its ability, has or has not lost its ability to hydrolyze GTP. So in this assay, we incubate radioactive GTP with the GTPase, and then look using thin layer chromatography at the amount of GTP and the amount of GDP that's present after certain time periods. So the Q61 mutation, the codon 61 mutation, there's basically no hydrolysis of GTP. The wild type proline 29 and phenylalanine 28 have an ability to hydrolyze GTP. And interestingly, P29 and F28 maybe have a little bit faster ability to hydrolyze GTP than wild type. Probably this is not physiologically important, because when you add a GTPase activating protein, a gap, this rate increases very dramatically. We also checked the cellular phenotype of P29S, and the upshot here is that P29S is localized to membrane ruffles in a similar fashion to F28L. Wild type is slightly different, and the Q61 associates with membrane ruffles and also has a large nuclear fraction, which has previously been seen for RAC. So the summary for P29S is that the traffic light, the important traffic light, is probably at this location. It's probably at the GDP-loaded form, whereby this is actually turned on. The GDP-loaded protein is actually able to release GDP and bind to GTP in a GEF-independent manner. And this, of course, causes increasing downstream signaling. So, summary for RAC is that this is the first frequently recurrent point mutation in a row family GTPase. This is the first frequent cancer-associated fast-cycling GTPase mutation. It's probably a different mechanism to the activating RAS mutations, codon 12, codon 61, and probably a different mechanism to the fast-cycling mutant at F28 based on the crystal structures. So the next steps that we're going to look at are what are the effects of guanine nucleotide exchange factors, GTPase activating proteins, and guanine dissociation inhibitors on RAC1, and also the potential impacts on this different conformation on downstream effector signaling. So that's the RAC story, and I will quickly go through the PAK story, and collaborators Ben Turk and Yossi have been collaborating with us on this. So what are the signaling pathways that RAC and row family GTPases signal through? They signal one of the guys they signal through the P21 activated kinases. And these guys are important for things such as proliferation, cell death, cytoskeletal dynamics, cell cycling. In cancer, there are alterations in PAK signaling, and those may be required for some oncogenic and metastatic phenotypes. Interestingly, however, the only PAK family member that is transforming when overexpressed is PAK4. So this and other data formed the basis of the Pfizer drug discovery effort for a PAK4 inhibitor, which failed in phase 1 clinical trials because there was basically no dose response. So what are these proteins? So these are a family of serine threonine protein kinases. That's a family in two types, the type 1 PAKs and the type 2 PAKs. The domain organization of these proteins is that there's an N-terminal region that includes a P21 binding domain or crib domain, a region that binds to GTPase, and a C-terminal kinase domain. For the type 1 PAKs, there's also an autoinhibitory domain. The big question that we've been trying to address in my lab is, well, how are the type 2 PAKs regulated? Because these guys don't include an autoinhibitory domain. And the regulation mechanism for type 1 PAKs is very well understood. There's a trans-inhibited dimer that becomes released on binding to the GTPase. GTPase binds the crib domain, releases the autoinhibitory domain, and you have an active kinase. Type 2 PAKs don't have that, and this is an unresolved question in the field. How are the type 2 PAKs autoregulated? So to address this, we generated full-length PAK4 and expressed this, and then conducted kinase assays. And kinase assays were with radioactive ATP, magnesium, and a substrate myelin basic protein. And we wanted to, first of all, address whether this protein could be activated by RAC or by CDC42, its associated GTPases. And we see basically no activity, no phosphorylation of myelin basic protein for the full-length PAK4. This contrasts with the catalytic domain alone. So we made and expressed and purified the catalytic domain of PAK4, and what we see is a dramatically different effects on downstream on phosphorylation of myelin basic protein. A lot more phosphorylation compared to the full-length protein. So we thought this was interesting. We then tested whether we could regulate the activity of the catalytic domain by incubating with constructs that encoded regions of the N-terminus. So we did these kinase assays with the catalytic domain plus regions of the N-terminus with myelin basic protein as the substrate again. And what we found was that some of these guys basically had no effect on catalytic activity, on phosphorylation of myelin basic protein. However, some of these constructs actually did have quite a significant effect and quite significant retardation of the phosphorylation of MBP. This we thought was interesting. It gave us hope that we could maybe see this mechanism by crystallography. So we obtained crystals of full-length PAK4 and the catalytic domain of PAK4, and we compared them, and we got these crystals in the same crystal form. The full-length PAK4 had a region of unmodeled electron density, and that was sat in the site of substrate recognition. This region of unmodeled or unsatisfied electron density was not visible in the catalytic domain alone, indicating potentially there's a region in the N-terminus of this protein that was causing this change in electron density. So based on the crystal structure, based on the kinase assays, we came up with two hypotheses for potential regions that density could be, the green and the red, and we obtained co-crystals of each of these peptides with the catalytic domain. The co-crystal of the green peptide basically showed no electron density at that particular location. The co-crystal with the red peptide showed a significant amount of electron density that was very, very similar to the full-length protein. So based on that, we made point mutations around the region of that sequence, which is close to residue proline 52. And those point mutations, for example, here and here, release the ability of these constructs to inhibit catalytic activity. So, for example, 1 to 130 when you have those point mutations, does not inhibit phosphorylation of MBP as much as the wild type. We also introduce those point mutations into the full-length protein, and we see we think a dramatic increase in the activity of the full-length protein when you introduce these point mutations around proline 52. You put this into cells, and there is an increase in cell rounding for the mutated PAK4 compared to wild-type PAK4. There are changes in the cytoskeleton, and there are increases in phosphorylation of downstream substrates. So we think this is an activated mutation. So what is this doing? So when we look at the crystal structures, there's a peptide that's bound in the substrate binding site, and this region is where the kinase recognizes its downstream substrates and phosphorylates them. There's a proline here. Proline 52 is sat right at the site of phosphoacceptance. So if you compare this structure to a previously solved structure by a group in Oxford with PAK4 with a known substrate, you can see that the phosphoacceptor site serine residue is actually replaced by proline. So we think this is the basis for autoinhibition of type 2 PAKs. So based on this and based on some other work we did, we have a model for type 2 PAK regulation. Type 2 PAKs are, actually one of the interesting things about type 2 PAKs is they are always activation loop phosphorylated in a cell. And that's been one of the conundrums here. Why is it always activation loop phosphorylated? Kinases are usually phosphorylated on the activation loop to change between the active and the inactive state. Turns out these guys can be activation loop phosphorylated because they're also inhibited by an N-terminal peptide sequence, an N-terminal sequence. GTPase binds, and binding of GTPase causes a translocation, takes the kinase to where it needs to be in the cell, and that's been shown by model groups. What we think happens is that there's an activating signal, potentially an SH3 domain, that binds and rips off this autoinhibitory substrate and allows the kinase to become activated. So, to sum up the PAK work, type 2 PAKs are regulated by an autoinhibitory pseudosubstrate, residues around proline 52 autoinhibit kinase activity. We think this explains why type 2 PAKs are predominantly activation loop phosphorylated, and we think there's an activator, potentially an SH3 domain protein that releases PAK autoinhibition, and we're looking for who that could potentially be. Okay, so the people who've done this work: Byung-Hak Ha, Matt Davis,